Okay. I hope these are coming okay. Again, refer to this, the PowerPoints along with these videos. I'll write down more explicit things, how we're going to move from here in terms of assessments. But let's just finish these lectures up. Okay. All right, the next type of structure that we're looking at includes fatty acids. So far we have covered that have the fatty acid parts. We talked about fatty acids by themselves, saturated and unsaturated. Then we talked about where they're found. First place with waxes. Okay, and then we talked about them found in triglycerides. And then we just got done talking about soaps. Okay, another place that these guys are found is in phospholipids. Okay, phospholipids are quite a bit like triglycerides, but instead of having 3 fatty acids, they have, instead of a third fatty acid, a phosphate group that essentially makes a structure very similar to soap molecules. By adding this phosphate, it turns a triglyceride, which is completely nonpolar and insoluble water, into being a soluble, partly soluble molecule. And that's because phosphates are essentially polar and will be attracted to water. So you're going to see similar structure that soap forms, that phospholipids could form as well. Okay. Here's a picture of the triglyceride versus the phospholipid. On the left is a triglyceride, and on the right is a phospholipid. You notice on the right, the phosphate essentially is like a polar head. And now what you have is a molecule that has 2 nonpolar tails versus soap that just have 1, and it has a polar head. All right, and here you go. Here's a picture of it. So you have the little green part is the phosphate part, making it polar, and then the orange parts are nonpolar. So the green part, purple part will interact with water, and the orange part will interact with itself, the nonpolar parts. Okay, all right, now why are these important? They are important because they form cell membranes. Okay. When you put a phospholipid into water, what it will do, it won't form a micelle, but what it will do is it will form a bilayer, where the nonpolar tails will line up next to each other, side to side and end to end. And that what their interactions between the tails is London dispersion forces, just like the tails in the soap molecules. And the polar parts will interact with the water through dipole-dipole. Okay. So here's a picture of what happens when you put phospholipids into water. Okay. So all compass, this is all happening in water. So when you put the phospholipid, what happens is they go tail to tail and side to side, increasing their London dispersion forces. So you have these 2 tails here will interact with these 2 tails here, end to end, but they'll also form side to side interactions. Okay. The other thing is that these guys here will interact with water. So these phospholipids will form bilayers. Okay, cell membranes are composed of 2 layers of phospholipids and phosphobilayer. And phospholipids are the main component of cell membranes. And so what happens is that when you put phospholipids in the water, they form a bilayer, where this hydrophilic polar interacting with the water, and the tails interacting with each other. And what happens is it essentially forms a sphere where has an inside, maybe this side here is the inside, and this side is the outside. So the inside has interact with the water of the inside of the cells, and these guys out here interact with the outside water. And there's a barrier of hydrophobic nonpolar tails that separates the inner water from the outside water. And therefore you have a place to have your cell mechanisms. When a cell, what a cell needs to do, needs to be by itself where it can do its own job, separate from the rest of the organism. Okay, so what it does is it has an encapsulation, and an encapsulation is formed by this phospholipid bilayer. And inside that water is where all the cell activity takes place. And on the outside is what's happening on the outside. And they're separate from each other. Now, one of the problems is this. You can't, the cells want to be separated from the outside world so they can do their job. But the other thing they need to do is they need to interact with the world when they need it. For instance, when a cell needs energy, it will open a door and let glucose in once insulin comes by and opens the door. So there has to be some interaction between the inside and outside world. So what we do is a cell is not just phospholipids, but it's a fluid mosaic model which incorporates many different types of molecules, not just the phospholipids. A cell is mostly phospholipid, but it also has other things too. There's proteins, carbohydrates, and cholesterol. And we're just going to go over a few of these. Cholesterol is I'll show a picture of this in a minute. Cholesterol is in the cell membrane to help stiffen it, to give it, make it more firm, not to have it floppy. All right, carbohydrates will stick on the outside, and what they do is they tell things on the outside of the cell who they are, what kind of cell they are. Like I'm a liver cell, and you can tell by the kind of carbohydrate I got stuck to me. There's other things called cell receptors for hormones and other things like neurotransmitters. Even the coronavirus has a cell receptor that attaches to. So there's something on the outside of your cell that this coronavirus recognizes, a cell receptor, and when it latches onto that, it lets that it enters the cell and makes more of itself. Okay. There are proteins. Most of the cell receptors are proteins, and we'll get into that when we talk about proteins in the next test area, test 4. But there's other ways that things can get in and out of cells, and those are transports. And we're going to go over those in a minute. And right now. Okay. First of all, this is a diagram of the fluid mosaic model. Okay, so the green are the phospholipid bilayers. All right. The orange is the cholesterols, and again, they're there to give it structure and rigidity. There's carbohydrate side chains that are used for recognition. There's things like these are the cell receptors, okay? And then we have these guys here, which are channels which let in things in and out of the cell. So this is the fluid mosaic model. All right. Now, I'm sure you will cover this more in depth in AMP, but we'll cover them briefly here since we're talking about cell membrane and its structure. So, how do things get in and out of the cell? Okay, first of all, polar things, which most things are in living, we are mostly water. Most things that are in our body are soluble in water, and they're polar. The problem is that, so this is good and bad. First of all, the phospholipid bilayer, because of the nonpolar parts, will not let polar objects through the membrane. So polar molecules will hit the phospholipid membrane, essentially bounce off, and then stay on one side. Okay, so the nonpolar center prohibits that from happening. But small molecules like oxygen and carbon dioxide, which are also nonpolar, will slide through the membrane and get into the cell. And that's a good thing because we need oxygen to move without resistance into the cell to help provide energy through cellular respiration. And we also need carbon dioxide to move out easily because of the waste product. But, however, there are some things that need to get in our cell that are just too big and polar. And how do they get in? Okay, so there's three different ways that we talk about things getting in and out of the cell. First of all, the one on the left is diffusion or passive transport, and that's like oxygen and carbon dioxide. They just slide through because they're small and nonpolar. Okay, some things will get help by proteins called integral membrane proteins, and these are kind of like a tunnel, and they'll let certain size molecules through, and they're very specific. They just don't let anybody through. They'll let in types of molecules. Okay, but it doesn't require any energy. Finally, the one on the right is what we call active transport. That's where sometimes the cell says, I need a certain type of molecule in the cell. I'm willing to pay for it. Okay, and so what it does is it uses up some ATP, which is basically currency for the cell, and then what they do is molecules can move in and out through active transport. Okay. So that covered the phospholipids, and we are now done with the fatty acids. I will go category. I will now spend the next videos on steroids, their structure and function.